Hey there, welcome to this explainer. I'm so glad you're here. Think of me as your smart study buddy today. We're going to break down this entire topic cleanly and clearly so you can grasp the core concepts really fast. Today, we're tracing the incredible journey of a product, from a flash-heated factory line all the way to a life-saving inhaler in a patient's pocket. It's this whole world of invisible guardians and extreme precision, and we're going to make perfect sense of it together.
Here's our quick roadmap. 1. What is aseptic processing? 2. Media fills, the dress rehearsal. 3. How to run a test. 4. Meet the validation team. And 5. Validating life-saving aerosols. Let's dive right in.
All right, Section 1. What exactly is aseptic processing?
Before we can understand how to test a sterile environment, we need to know how we create one in the first place. At its heart, aseptic processing is how a sterile product, like a drug or even food, is put into a sterile container without letting any contamination sneak in. To pull this off, the product is usually flash heated to some pretty extreme temperatures. We're talking 195 to 295 degrees Fahrenheit, that's 91 to 146 degrees Celsius. By doing this incredibly fast, we actually retain more nutrients and use way less energy compared to older methods like hot fill canning. This is the magic that keeps things safe for a long time without needing chemical preservatives. And in the pharma world, all of this is backed up by pristine clean rooms, special bacteria-retaining filters, and dry or steam heat.
Now, check out this exact breakdown of the sterile packaging used to hold these products once they're flash heated. You might think these containers would be made of super heavy-duty, complex materials, right? But it's actually really elegant. 70% of a typical aseptic package is just paper. Then you've got 24% polyethylene, that LDPE plastic, and just a tiny 6% aluminum. This exact combo is lightweight, but it creates an absolute impenetrable fortress against outside contamination. And just to give you a sense of scale, we aren't just talking about tiny juice boxes or little medicine vials holding a few fluid ounces. This tech scales all the way up to staggering 8 million-gallon aseptic tanks. Literally, these massive 30,000 cubic meter containers are put on ocean-going ships, utilizing this highly efficient bag-and-box technology to move millions of gallons of sterile liquids around the globe. It's wild.
Moving on to Section 2, Media Fills, or, as I like to call it, the dress rehearsal.
So how do factories actually test these highly sterile environments without wasting a ton of real, expensive medicine? Enter the media fill. Think of a media fill, some people call it a process simulation, as the ultimate theatrical dress rehearsal. We absolutely need to know our manufacturing lines are flawless. But instead of using the real active drug solution, which would be a massive waste if something went wrong, we run the whole process using a sterile microbiological growth medium. Basically, it's a specialized broth. If any microscopic germs somehow crash the party because of human error or a machine flaw, they will thrive in that broth, and we'll catch them before real drugs are ever produced.
It's a brilliantly simple swap. In routine production, everything is live. You're processing the actual drug solution that's going to patients. But in a media fill, everything looks, feels, and operates exactly the same. You've just swapped the real medicine out for that growth medium. It's a simulated run designed specifically to hunt down contamination errors safely. The primary goals for this dress rehearsal are pretty straightforward. First, we evaluate the aseptic assembly and the equipment. Second, we qualify the human operators to make sure their technique is spotless. And third, we demonstrate that the environmental controls of the clean room itself are fully working. But here's a massive caveat you should definitely commit to memory. A media fill does not validate the filter's own ability to sterilize the growth media. It's testing the overall process, the environment, and the people, not the filter itself.
Okay, Section 3, how to actually run a test. Knowing exactly how to run this test properly brings us to some very strict rules. Before an operator is ever allowed to touch the real product, they must undergo initial process qualification. Obviously, they have to already be fully trained in gowning and handling sterile materials, but to become fully qualified, they have to pass the Rule of 3. This means executing 3 complete media fills, and they must do it on 3 separate days. Plus, no generic testing is allowed. They have to use the exact production process at the specific facility being qualified. And the testing doesn't stop once you pass that first time. You can't just rest on your laurels here. To keep that elite, qualified status, you have to successfully run at least 1 media fill every single year. If anyone fails, or if there's any sign that the line didn't maintain product sterility,
all those media fill validations have to be conducted all over again.
When designing the test itself, you need absolute precision. Every single downstream step, from the sterilizing filter all the way to product release, has to be simulated. After the containers are filled, they're incubated to see if anything grows. And here is a highly specific rule for the prep. When making the growth medium on site, do not use filtration to sterilize it. Instead, you're supposed to use a validated process like a steam autoclave. That ensures the broth is perfectly sterile before the simulation even begins.
Section 4. Let's meet the validation team.
So who is actually calling the shots and enforcing these strict dress rehearsals? It takes a multidisciplinary powerhouse team. You've got the head of quality assurance, the head of engineering, the validation manager, the production manager, and specialized discipline experts. This team writes the site's master validation plans, figures out exactly what equipment needs testing and how often, and makes sure the testing protocols actually prove the process is safe. They're basically the ultimate gatekeepers of quality.
And they don't just validate for the fun of it. This team springs into action during very specific triggers. We're talking a totally new process, brand new equipment arriving on the floor, or when an existing setup is altered. Basically, if standard end product testing is deemed a poor indicator of quality, this team steps in to run extensive simulation trials and worst-case scenarios just to guarantee the final product stays absolutely pristine.
Which brings us to our final section, Section 5, Validating Life-Saving Aerosols. Let's anchor all of this rigorous theory into an everyday reality by looking at a daily life-saving application, aerosol inhalers. Since the mid-1950s, inhalation aerosols have been delivering drugs directly into the respiratory system. The most common one is the metered dose inhaler, or MDI. You know the devices people use for quick relief from asthma, emphysema, or COPD? Well, that intense pharmaceutical validation we just learned about, that is exactly what underpins the regulatory compliance for these MDIs, which ultimately improves the quality of life for millions of people. Here's a quick breakdown of the anatomy of a pressurized MDI. You have the canister holding a mix of the drug and the propellant. The plastic holder is the body of the device itself. The propellant is a liquefied gas that actually shoots the drug out. But the star of the show is the metering valve. It's meticulously validated to make sure that the exact, perfect dose of medicine and gas is expelled every single time you press it. And of course, the mouthpiece is where the patient breathes it all in.
But here's the fascinating human catch to all this perfect science. You can have flawless factory validation and aseptic processing, but the patient still has to use it correctly. It actually requires a person to perfectly coordinate their exhalation and inhalation with pressing the device. If your timing is off, the medication simply won't reach your lungs properly. Thankfully, you can attach these bulky spacer devices to the inhalers, and that helps solve the coordination problem, making it way easier to use outside the home.
So we've covered a ton of ground today, from the factory floor all the way to a single breath. I want to leave you with this thought. Next time you use an inhaler, or even just pour a glass of juice from an aseptic carton in your fridge, ask yourself, just how many items in your own home are secretly guarded by these intensive, invisible dress rehearsals? It's pretty amazing to think about. Thanks for learning with me in this explainer today, and keep asking questions about the hidden systems that keep our world safe.